all of you. I think most of you know Nick from a number of meetings. So I'll just give you a quick introduction to his CV and show you maybe one slide. So Nick studied medicine in the University of Geneva and received his MD, PhD, both his MD and his PhD degree in 1993 in cell biology and in medicine. And he then went on to do a postdoc with Sergio Grinstein in Canada and at the Hospital for Sick Children. I think that's where he started his career working on sodium proton exchangers. And his research since then has very much focused on mechanisms controlling calcium and pH-D homeostasis. And he's really focused on innate immune cells. And we will hear some of his research. So after his postdoctoral stay, he went back to the Department of Physiology in Geneva and has been there and proceeded on from associate professor to full professor and in the Department of Cellular Physiology and Metabolism. And I'd known of Nick's work for quite some time, but let me just see if I can share my screen now. No, this doesn't seem to. Oh, okay, here. Let's see if this works. I don't know if you can see my screen. Yeah. So can you see my screen? Actually, somebody has to say something.
Yes.
Okay, great. So anyway, so I've known of Nick's work, but we hadn't really met much until the FACEP meeting in 2016, which took place in Lisbon. And it's so nice to see these in-person meetings now that we're all stuck staring at our screens. And at this meeting, kind of on a spur of a moment thing, Nick and I decided we were actually elected to co-chair the next FACEP meeting, which then took place in 2018 in Lake Tahoe in California. And that was a meeting that Murali had organized. It was a very exciting calcium meeting. And here you can see, and the nice thing about a real meeting is that you get to do some fun parts besides just listening to talks. And so we went, the whole group went kayaking, and we had lots of scientific discussions. And Nick actually saved my PhD student's life almost, because one of the kayaks got missing and Mike jumped into the water and got hypothermia, and so that was really fun. And then we met again at another meeting. This is in Morocco, in Fez here, and you can see Don Gill and Indu here, and Beth, and a lot of the calcium gurus here. And then we saw each other again at a meeting in India. So it's been really fun, these real meetings. And so we're kind of really sad with the current situation that the FACEP that we were going to share was supposed to take place July 5 and 10 in Dublin, and that was cancelled. Then we tried to move it to this year, July 15 and 20, but due to Corona, that's cancelled again. But Nick hopefully will tell you something at the end of his talk that when we actually might meet again in person. So it's great to have the webinars, and I'll stop sharing now. Yeah, it's great to have the webinars. It's great, but I'm really looking forward to see all of you guys again in person to discuss science. It's just so much more lively, and to discuss science also while taking a hike or going kayaking. Okay, I think that's all my introduction to Nick, and go for it.
Okay, thank you, Barbara. So I guess you guys hear me well? Yes, okay, so just for the record, for this kayaking incident, your student was actually diving in freezing water to get a kayak that I had mistakenly let loose. It's actually my fault on this thing. So I'll share my screen, and it's really nice to see you guys, those of you who turn on your video. There, you should now see my screen. That okay? Barbara?
Looks good.
Okay, looks good. So before starting this presentation, I have to thank a few people. So Martin contacted me, but I think it was Katja who actually remembered me at some point. So Katja Ritzdorf, thank you for all the time and effort you spend in organizing this. You're not alone. There is also Malène Orhus, who's there, and Emily Cood. I think she's the person behind all the IT. So all these things are running really smoothly. So thank you very much for all your time and effort. So I have chosen a new set of data because I feel that you guys have seen my phagocytosis business too much. So this is something different. It's about the acylation or palmitoylation of the ORI channel, something that we started a couple of years ago and that got a really good boost with the incoming in the lab of Amado Carrera Sureda, who's a postdoc who's also attending the meeting. So he put this story out. I chose to start by remembering that this is now 35 years since Jim Putney, who's in the audience, hello Jim, formulated his hypothesis about what was called then the capacitative calcium entry. So you can read here in verbatim is a very nice text that was published in Cell Calcium. All this is still true. So model proposed for the mechanism by which the activation of the surface membrane receptor activate this sustained entry of calcium into the cell here. And the hypothesis was that when the intracellular pools are empty, and this happens when you add an agonist and you open this IP3 channel, you empty the pools, a pathway from the extracellular space to the pool is opened here. So this is this capacitative hypothesis. And conversely, when you refill the pool, the pathway is closed. And this has really stood the test of time. Now, 35 years later, you find it in textbooks. And so this is one a bit more elaborate scheme from Murali and Rich. And you see here all the players. So the receptor coupled to IP3 releasing calcium from the pool, which is mostly the endoplasmic reticulum. And then you have this pool talking to the store-operated entry channels. And you can empty the pool by different manipulation. We use tachycargine. Many people do that. You can also use ionomycin or calcium chelator like TPEN. Or the people who do the patch clamping, they use BAPTA or EGTA to chelate the salicylic calcium and basically suck out the calcium from the pool. And this is leaving via this calcium leak pathway. Any of those mechanisms that would empty the ER would activate the store-operated calcium entry pathway. And this has been verified countless amount of time. You can do it by microscopy. This is a tachycargine response in calcium-free. All of you know that. You re-add your external calcium, you see this increase in salicylic calcium, and we call this SOCI. You can measure the slope, the amplitude, or the integrated response. Variation of that, which we like in the lab, is to use the manganese quenching technique, where we add manganese, and then you can deplete the pool either before or after adding the manganese. The entry of this ion will quench the Fura2 fluorescence, and you see actually you can follow the rate of quenching, which is a pretty good surrogate of the flux across the membrane channel. Now, this methodology was established already in the 80s. In the 90s, people managed to record these currents. So Rich Lewis contributed, Reynold Penner, Mike Allen. So they recorded this channel, which they called iCrack. So I must confess, I don't like this name. Being medical doctor, I don't like to use drugs names and to label something. So anyway, this has stood. So the electrophysiologist will call it iCrack for current calcium release activated current. And this is its typical kinetic of activation when you rapidly deplete the store with IP3 or with ionomycin. And this is the typical current voltage relationship that you get by applying voltage ramps from negative to positive voltage. So this steep inward rectification is a characteristic of the channel. Now the molecules, they came later. They came in the 2005, 2006, and you know all about these two molecules, the STIM and ORI family of proteins. So just briefly, this is the activating cascade. So the two proteins have to come together for the channel to be active. The channel is the ORI family of calcium channels. This could be ORI 1, 2, or 3. And activation of STIM is triggered by the decrease in ER calcium concentration, which basically causes an inside-out conformational change. The STIM exposing these residues, polybasic residues, that will basically bind negatively charged lipids on membranes, mostly plasma membrane, but not only. I've been studying the phagosome, so this happens also on internal membrane. And at these sites, two things happen. The binding of STIM will generate these membrane contact sites. It will bring these two membranes in close apposition. And this residue, the CAT domain, will trap inactive ORI channels and activate via direct binding of this CAT residue to the C-terminal domain of the channel. And you can actually see the morphological changes that are occurring in the cell by this interaction. So this is what happened to the ultrastructure. If you image a cell, this is a HeLa cell, you see that the endoplasmic reticulum will elongate and form this very thin cortical structure, which we dub the thin cortical here. And these are really enriched in STIM protein, which we can see by immunogold here. So those are the morphological change. You can also see this mobility of the STIM and of the ORI channel by turf imaging or fluorescence imaging, because they will make this nice punctate structure. Now, in the last decade, I think the research is focused on the molecular and structural changes. So we had structures of the Drosophila ORI, we have partial structure of the STIM, and this is what is now, to my knowledge, at least the current models for the activation of STIM. So the first step during calcium depletion, the unbinding of calcium from this EF end will call this basically zipping of this cold coil, which in turn will unzip this other cold coil here that release this calcium channel activating domain or SOR. So this is the change in STIM. In the ORI, it's a bit more complicated. So there is a model from Murali proposing that there is a rotation in the pore that basically lets the calcium flow. And from the structure of the closed and open Drosophila channel, there is a postulation that there is a dilation of the pore which opens when this helix is bound to STIM. So that's about my knowledge on the STIM and ORI. I know about acylation or palmitoylation. I have to confess I knew very little when I started. So I had to read a bit. So this is post-translational modification. And it's the only reversible lipid post-translational modification. You have other lipid modification like meristylation or farnesylation that are irreversible. But this one, which occur by a thioester linkage on cysteine residues, typically involves the addition of palmitate, which is a 16-carbon chain lipid. And this one is reversible. It's catalyzed by two family of enzymes. The addition of palmitate is catalyzed by palmitoyl acyl transferase. They have a terrible name. It's ZDHHC. So in the lab, we call them PAT because it's easier. There are 23 isoforms of these in humans. Typically, they are transmembrane protein with the catalytic sites exposed on the cytosol. So they would add the lipid. And the reverse reaction is catalyzed by the acid thioesterases, of which there are two isoforms in. So with these two opposite reaction, a bit like phosphorylation and dephosphorylation, you can chemically modify proteins by changing their lipidation status. And as you can guess, this is a lot of functional implications. So from a review of like Gibson, you can see you can control pretty much all the steps in the life cycle of an ion channel, from the assembly, maturation, sorting, and then membrane insertion. Interestingly for us, distribution in membrane lipids, and also a retrieval from the membrane and degradation. So all these steps can be controlled by changing palmitoylation of proteins. And for transmembrane protein, there is another layer of regulation, because the addition of lipid can have multiple impacts. For instance, here, if you would palmitoylate this tail, you could anchor it to the membrane, and that could bring this protein in close association with a partner. So you can increase protein interaction by this mechanism of palmitoylation. Similarly, you can prevent it by just removing the active sites from the binding. Okay. And now, this addition of a lipid to a chain of a transmembrane protein can also change its distribution in membrane lipids. So it could typically favors insertion in larger or thicker, more ordered lipid domain. And that can, in some instances, resolve hydrophobic mismatches between in proteins. So it will allow it to go there, while its structure, in theory, should not allow it. And it could do it just by inserting, or also by imposing a tilt in the transmembrane helix. So you can have actually pretty big structural change in transmembrane protein that could be conferred by this post-translational modification. So just to finish the introduction, 2 examples of known transporters that are modulated by palmitoylation. The NCX, which indeed I studied at some point in time. So recent findings showed now that the palmitoylation of the NCX1 does 2 things to this protein. It will target it to lipid rafts, so more ordered lipid domain rich in cholesterol. And it will also change the access of the inhibitor to the binding site. So the two properties that would be important for the activity of this transporter are modified by 1 step of post-translational modification. And for the P2X7 receptor, there is structural data now that show that the addition of palmitate to the pore lining helix here will basically anchor it to the membrane. And this process completely prevents actually the desensitization of this receptor channel. So this is a pretty impressive modulation that is conferred here by the palmitoylation. And now to go to our focus, I got a little bit of geographic advantage here, because next to Geneva is Lausanne, and I have a colleague there, Gisou van der Hout, and she's working on palmitoylation. And we meet from time to time, and she's maintaining a database of palmitoylated protein, a resource, which I recommend you use if you're interested in palmitoylation, or if you want to know if it concerns your pet protein. And she told me, well, you know, your ORI channel is palmitoylated. I said, oh, okay, interesting. And it's palmitoylated in the 2 species, mouse and human. 2 of 17 palmitoyl proteome have reported that the ORI is palmitoylated. And for the human, it's found in T cells and platelets, which are cells in which the ORI function has been established, and where you have an impact. You have actually disease in humans that are caused by loss of ORI in these cells. So that got us started. And since she has the knowledge to study palmitoylation, we started a collaboration. And basically, she did that for us. Laurence Abrami in her lab did that for us. They looked at the accumulation of tritiated palmitates into the ORI channel. So you just incubate the cells with this tritiated palmitate, and at some point you add this hydroxylamine, which breaks the thioester bond, the acyl thioester bond. And you see here, you have to expose these blots for like 3 weeks or 4 weeks to see a signal. You see in this autoradiogram that the ORI channel is palmitoylated, and you can prevent this by adding this hydroxylamine. An independent assay was used here, in which you first treat with the hydroxylamine, and then you add a PEG molecule, which is a known molecular weight, which will change the mobility of your protein on a gel. And you see here that you can pegylate this endogenous ORI in HeLa cell by treating the cells with this HA and then adding the PEG. So when we had this knowledge, we looked where in the channel could that happen. So good news is only 3 cysteine residues in the ORI. 1 is outside, so very unlikely to be palmitoylated. So we were left with these 2. They are both in the second transmembrane domain. 1 is close to the cytosolic site, so accessible. The other one could, in theory, be palmitoylated. It happens sometimes inside the membrane, but it was less likely. The other hint was that these 3 residues are very well conserved, but the 143, which is the one that is just at the beginning of the second transmembrane domain, is completely conserved up to C. elegans. So that was already our top candidate. And what we did is generate mutants. Monica Bulla in the lab generated those, so mutating the cysteine to either alanine or serine, and then exposing these gels these cells to tritiated palmitate. And you have here in 2 cell types, HeLa cells and RPE cells. And we lose the signal in the C143A mutant, or in the double mutant, but not in the C126A mutant. So this tells us that this ORI channel is chemically modified by S-acylation on cysteine 143. So at this stage, we had a residue, and we were looking now for an impact. So we started with these HEC triple knockout cells, HEC293, provided by Maud Trebach, I believe, and restored the signal here by re-expressing the different mutants. And you can see that the mutation at site 143 reduce the store-operated entry, while mutating the 126 does not. Okay, and the aggregate data are here. And then Amado came, and he made stable cell lines. He likes very much this system. So he made cells stably expressing MTI STIM and ORI GFP, sorted them for similar fluorescence, as you can see here, and then looked at the function, and here they are the data. Of course, now you have both STIM and ORI, so you have stronger SOCI, but the results remain the same, mutating the 143 residues, either in the single or in the double mutant, have a strong impact on store-operated entry in this cellular system. So now we had the chance to have in the lab an electrophysiologist, Ji-hee Kim, coming from Korea for 1 year stay here. So she managed to record the currents from these stable cell lines. So here are 2 example traces of the activation of the current, this iCrack, and the typical profile. And here is the aggregate data. You see that there is a reduction in current density, which pretty much matches what we see in the microscopy. There is also a delay in activation. The current will activate more slowly in this system. I'm going to show some data that were requested by reviewer. They say, okay, it would be interesting to know about fast calcium-dependent inactivation. And we were surprised because we were not expecting these to be modified. So these are current evoked by short voltage steps, ranging from -120 to -60 millivolts. And you see here the typical rapid inactivation of the current, which to see you need to reduce the buffering power, so moving from 10 millimolar BAPTA to 10 millimolar EGTA. And as you can see, the kinetics of this and the amplitude of this is reduced, and you have also here, which is known as a reactivation. So if you graph now this, the fractional current which remains at the end of this pulse, this inactivation is quite reduced in the C143A mutant, and the kinetic of inactivation is accelerated, as you can see from the trace here. So that was a bit unexpected for us. We were looking for possible mechanisms for this, and a typical feature that people observe is clustering of the channels. So for this, you just treat the cells with tapsicargine and image them on a turf microscope. And this video shows, the top one shows the wild type ORI aggregating following tapsicargine addition. Same in the bottom panel. Looks pretty normal at first glance, so they can make clusters. Here are blown up images of the clusters. These would be the STEAM clusters. Here, these are the ORI clusters. And on close inspection, it appeared to us that this cluster would be smaller, appear to be smaller in the ORI mutant. So Amado looked at that systematically, and here are the time course of cluster formation for both ORI here and STEAM here. So number of cluster is identical. The fluorescence, the overall increase in fluorescence for STEAM is normal, is indistinguishable between the two. For the ORI, you don't see an overall change because it's basically membrane rearrangement. But the size of these began to differ after a while here. So in the steady states, when you look at the steady state, we have a clear decrease in the size of the individual cluster, which will translate into a decrease in the total area or percentage area of the membrane which is covered by clusters. So we believe that this is a true defect of channel assembly, and it's not due to difference in membrane expression, at least at this stage, because if you measure the percentage of the fluorescence in the turf plane relative to the total fluorescence, there is no difference between the two conditions. So we have a channel assembly defect. Then he looked at channel mobility in the membrane. For that, you need to use this FRAP technique where you photo bleach your dye and you look at the recovery time. So these are example images of the ORI GFP in the wild type cells. You bleach and then you see the fluorescence coming back. These would be the individual recordings from the two conditions. And there is a slight but significant difference in this mobility. The mutant will diffuse more slowly. So this will be expressed by the kinetics. But the percentage of mobilizable protein is the same. So the mobile fraction is the same in the two conditions. So different mobility in the membrane. Then Amado wanted to know whether this different mobility and different clustering was not to distribution in different lipid domains. So here was an interesting reading of papers, mostly people doing palmitoylation, to see how do they study that. So what they do, they actually do these giant plasma membrane vesicles. So for that, you have to basically use a buffer that makes your cell bubble. And then you collect these cell blebs, you adhere them in the cold. And in the cold, if there is a mixture of lipids, the lipids will dissociate. Okay, so you will have segregation of the two lipids. It will look like that. So this will be your protein of interest, in our case, the ORI GFP. And you see that there are patches in the membrane where it accumulates. This would be the staining, it's actually inverted, sorry. This would be the staining for PIP2 cherry in red here. And this would be the staining with cholera toxin 647. So this is a typical Raft marker, and this is a PIP2 marker. And to measure this accumulation, you draw a line between a region that is rich in a given marker and an opposite pole which is poor, and then you take the line's profile and basically measure what proportion of the protein is in the rich or in the poor region. So this is still ongoing. The images with the wild type and the mutant look like that. It seems that the mutant is more accumulating with preferentially in PIP2 rich domain and less so in cholesterol rich domain. These data at this stage are still not significant, but Amado has just started to redo that using an improved procedure. So I hope we'll continue, and with some modification, I think there is something interesting to follow here. No, once we knew that we had a palmitoylated protein, the next thing to know for our colleagues in Lausanne would be to know, okay, what is the enzyme which is modifying your channel? They had already a list of candidates, which you see here. They did not test all the 23, but they tested what they believed would be the most promising candidate. And quite systematically, they saw that 3 of those enzymes, isoform 3, 7, and 20, would promote the palmitoylation of the ORI protein, both in these RPE cells and in HeLa cells. Okay, so we immediately check whether removing those enzymes will have an impact on the function of the channel, and it does. Actually, if you silence these 3 enzymes, you have a pretty big loss of function of the ORI channel in our cell line. But conversely, when you overexpress, which was surprising, only the 20 isoform would cause a gain of function. They were not too surprised, our colleague, because they know that this PAT20 is actually preferably associated with plasma membrane protein. So they say, okay, yeah, this is, to them, was expected. The reviewer, they don't like it, that it's not congruent, the 2 data of silencing and overexpression. But anyway, that's no problem. What is clear is that this PAT20 gain of function is seen in different cellular systems. You see here on top, these are HeLa cells. In the bottom, this is the stable cell line from Amado. And most importantly, if you use it in the cells stably expressing the C143 mutant, you lose this effect. So it's pretty clear to us that this enzyme, this isoform PAT20, is modifying the ORI channel on this residue 143, and that this causes a gain of function. Now we move finally to a different system, which is the T lymphocytes, and we started with the Jurkat T cell. And I'm showing here recent data about the impact of expression of these palmitoylation enzyme in the Jurkat, and it's like 100% congruent with what we had in HeLa and RPE cells. As for 3, 7, and 20 promotes palmitoylation. If you knock them down, 3, 7, and 20, you lose the signal. So what we see in HeLa and RPE also happens in the Jurkat. And of course, was interesting to know what that would cause to the function of these cells. So to study that, Amado engineered ORI knockout Jurkat. So he knocked it out by CRISPR. And this is the FACS profile of the fluorescence response to a tapsicagin readdition protocol. And then he reconstituted those cells with the 2 versions of the channel, the wild type or the mutant, expressing GFP and match for fluorescence level. And these are the recordings that we got on the microscope with these cells. So you have here the cells that has been CRISPR, not CRISPR'd, it's a control, okay? You remove ORI, you lose quite a bit, but not all, because there are other isoforms of the ORI in the Jurkat, but you lose a pretty big amount of the store-operated entry, which you can regain by re-expressing the wild-type channel, and you don't regain as much by re-expressing the palmitoylation deficient mutant. The aggregate data are here. And now these cells were up to be characterized. So to remind you, in the DURCAT, one of the main signal which is carried by the ORI channels are the activation of the antigen receptor, which will cause the proliferation of these cells. This involves generation of IP3, calcium release from stores, activation of the STIM-ORI pathway. The combined calcium signal will then activate calcineurin, which will cause the translocation of the NF80 to the nucleus, which will then activate transcription and the production of cytokines and induce the proliferation of the lymphocytes. So to our surprise, when we activated this pathway, we activated the T cell receptor using beads coupled with these with the CD3 and CD28 antigen. This is the normal response that you see, which is basically due to the IP3-induced calcium release caused by the ligation of the receptor. This first response, which is recorded in calcium-free, was already pretty much impacted in the cells reconstituted with the mutant channel. And now when you re-add calcium to measure this mixture of store-operated entry and what people used to call receptor-operated entry, you see here that there is a very strong functional defect in the mutant, and the aggregate data are here. So this was a bit surprising, but this translated into a very almost absent translocation of the nuclear factor of activated T cells. This would be addition of the beads and measurement of the nuclear translocation of NF80 would be severely compromised in the mutants, and so was the secretion of IL-2 cytokine, whose production is driven by the NF80. So in this cell line, there is a big functional defect which is caused by the replacement of this residue, which prevents palmitoylation of the ORI channel. So finally, to characterize these cells, we wanted to look at what the immunologists call these immune synapses. And this is basically the contact between an antigen-presenting cell and T lymphocytes. You can mimic it by adding a bead coated with antigen that will ligate the T cell receptor. So this is a bead which is coated with CD3. And what you see here is the fluorescence of the GFP tagged ORI channel. And on the bottom, these images is the actin structure that are labeled with this probe SIR actin. And you see that when the cell make contact, they will make these pseudopods that are enriched in actin. So this would be a kind of immune synapse. And this is what happens when we do the same with the mutant cell line. We see that there is less actin structure that are recruited here. And this would be the blown up images. And it turned out to be difficult to evaluate, in part because the bead here is also fluorescence, and the fluorescence overlap with that of the ORI. So when you do a line profile, which people normally do to see the accumulation of the actin, you do see it. This little increase here is basically the ORI at the cup, which you see here in this structure, and that you do not see in the mutant. But it proved to be too difficult to quantify because also these are very dynamic. So we turn to a different system, which is plating the cells on an activating cover slip, something we also use for phagocytosis. So basically, you coat your cover slip with anti-CD3, and then you let your cells adhere. And when they touch, they will make this immune synapse or this pseudo-immune synapse, and you can follow it by recording the fluorescence of the actin and of the ORI, which is shown in this movie. And to know where the T cell receptor would be located, what Amado did, he added TCR-PE, antibody against the T cell receptor labeled with PE, at the end of the experiment. So the synapse is already formed, and just to label it, to know where the receptors are, he adds the dye at this point. So here is the movie. So this would be the actin, and now you see appearing the receptor. So I'm going to play it again. The ori is here. Actin structures with the typically empty central structure, which would be the synapse, and which would be where you would expect the T cell receptor to accumulate. The ori channel have been reported to be recruited to the synapse, but this is still controversial. So I didn't realize when we did that we will enter a debated field, but anyway, Amado quantified all these structures. So measuring the number of synapse or pseudosynapse that form on these cover slips, the first striking observation was that the mutant cell would make a lot less. So the data are here. So about more than 200 cells were scored, and you have a clear difference in the number of cells that are able to make a synapse on this cover slip, depending on whether or not they express the mutant ori. The other striking observation was that the distribution of the ori GFP was different, and most surprising, the labeling of the T cell receptor was completely altered in the mutant. So this would translate into a strong signaling defect, which Amado also documented biochemically here by measuring the phosphorylation of ERK in the two cell line. So here are the aggregate data for the amount of ori GFP. There is a lot less which is recruited into the synapse, defined on the basis of the actin ring, so measured only in cells that form actin rings. And the number of these TCR dots, or this TCR staining, is really almost, not completely, but very strongly impacted by the mutation. And this is not due to a difference in expression of the T cell receptor in the two cell types, which is preserved. So to finally study these, Amado measured also the geometric parameters, so the size of those synapses that would form in the cells that form them was normal, but obviously since there are a lot less TCL receptor, the co-localization with the ori would be strongly impacted. And when he looked in different regions, so people will have different names for these different domains of the synapse, he would see that by removing preventing the palmitoylation of the ori would basically force the channel to accumulate in the central region of the synapse, which is depicted in this cartoon here. So in the wild type condition, you will have an accumulation through this zone, pretty even, but in the mutation, the ori will stay in the middle, and the T cell receptor would be depleted from the whole region. Okay, I think I'm pretty much done. Sorry if I run over time. So in conclusion, I hope I convince you that we show conclusively that the ori channel is chemically modified and functionally regulated by this palmitoylation or S-acylation. It's pretty clear to us that this occurs on cysteine 143 and that the PAD20 is critical because it confers both a gain and loss of function and modulates the store operator entry. The mechanism, we believe, may involve, at least in part, it may involve an alteration of the channel mobility and partitioning in the lipid. So we think that it may promote the channel insertion into lipid rafts. And from our data with the DURCAT, we think that this acylation is important for efficient immune synapse formation. And in that way, it will control the availability of the ori channel at the immune synapse. And here is the same in a graphical abstract that every journal requires. Welcome to see it hopefully shortly if we manage to publish this story. And I have to acknowledge Amado, who is in the audience, and did pretty much all the data with the Jurkat, the vesicles, the turf frap. Guizou and Laurence in the neighboring town of Lausanne, they did the biochemistry and measured the palmitoylation. Jihi, supervised by Maud, did the electrophysiology. Monica and Cyril and Jennifer. Monica started. Cyril is our morning technician. He's not online. He's probably already sleeping now. And Jen will help us with the biochemistry, which is required by our nice reviewer from eLife. And you can read this preprint version of this, which is a bit different. You can read the preprint on this site. So thank you very much for your attention. And I think it's up to you, Barbara, to handle the questions.
Yep. Yeah, thanks very much, Nick. That was very exciting. And there are already a couple of questions in the chat area, so you can raise your hand or write something in the chat. And I think Colin Taylor will start with the first question.
Should I close the presentation? Or?
No, I think we might need to go back to something or so. Colin, do you want to, should I read your question or do you want to just phrase it yourself? Colin? Well, Colin Taylor asked that he might have missed it, but how far can you go, e.g. with analyzing functional consequences of losing PET to demonstrate that phenotypes arising from mutations of C143A are due to loss of palmitylation rather than the mutation per se? And that's actually a question I also had. Did you try to mutate it, say, to a serine, which would be more close from the structure to cysteine?
Yes, we did. Actually, I showed in the early part, I showed some data with the serine mutant, which is to us undistinguishable from the, let me see, from the, here, this guy here. These two are serine mutants. So in terms of reducing SOCI, it's similar. In a patch clamp, they also are similar in terms of reducing the current amplitude, maybe even a bit better. So that's all I can say. No, it's true that most of the data we have with the mutation, but we also have the enzymes, so we can overexpress or knock down the enzyme, and that is a clear consequence on the function of both, at least SOCI. We have in HEC, we have in Jurkat. Okay, so we may also do something with this immune synapse business, overexpressing the enzyme. The drugs, the chemicals are not so good. I mean, there are some dirty inhibitors of this palmitylation reaction, but specificity is questionable.
Yeah.
Well, I'd rather stick with the overexpression or silencing of the enzyme.
Right.
I kind of like the overexpression of PAP20. Because as you have seen, the knockdown also does of the other isoform also reduce SOCI, and that may also be because of target effect. It may be interesting to you to know that the stem is also in the Swiss palm, so it's also palmitoylated. So we started with ORI, but there may be interesting findings to do with the stem.
Oh, cool. Gert has a question. Gert, do you want to?
Yeah. Hello. Sorry that my camera is not on, but great talk and to you, Nicolas, and great work, Amado. So are these enzymes constitutively active or regulated by other factors? And then related to that is that the palmitylation, could it be changed during agonist-induced calcium signaling?
I would think yes. I mean, there is reports that some are regulated by calcium, so it may very well be, and it's a very dynamic process. So it may very well be that you have multiple steps of palmitylation and depalmitylation, and some may be regulated by calcium. So yes.
Thank you.
Shmuel has a question.
It's about the same thing, is whether you see is any evidence that the native ori is actually palmitoylated, and do you see any change during T cell activation or something? I know it's difficult to address, but it might be interesting to see if there's something, or even from other cells or other palmitoylated functions, that they are actually a regulated process.
Yeah, well, I showed the native ori in the HeLa cell, so that was not an overexpression, this first finding here. So this
is the native.
This was the native ori, yeah.
Oh, okay, okay.
This guy here. No, in the Jurkat, what I showed, those was the cells stably expressing the GFP ori. So in the Jurkat, I don't know yet with the native. But we know that enforcing the PAT20 will change the function.
Thank you.
Since your phenotype was more dramatic in the Jurkat cells, do they express a lot more of the transfer enzymes compared to HeLa cell?
I don't know if it's more. I mean, the SOC is not really more dramatic.
Well, with the.
If you look, it's about, it's not that, but it's also because there is a remaining stop entry in these.
I was thinking more of the effects on end-fed translocation.
Yeah. So what was really striking to us was when we looked at this agonist evoked, TCR evoked signaling, that there the defect was really big. So it's a different thing. So it may require assembly of the channel in specific domains, which you may not need for when you're tapsicarging.
Antonio has a question, right? Antonio Villalobos?
Yes. Let me see. What I would like to know if there is any naturally occurring mutation associated to disease affecting that particular system.
Not that we could find, no.
Okay. Thank you, Nicolas. Great talk. Sorry, I must leave.
I actually have another question. I don't understand this TCR thing, because the TCR is upstream of ori, right? You'd get synapse formation and you'd get activation of the T cell receptor. So why would that be so dislocalized with altering ori?
So from my understanding, when you make the synapse, as you see, it's a dynamic process. So you have to remodel the actin, generate the structure. So it's a dynamic thing, and the calcium signal at this particular location is important. So Michael has shown that the ori is recruited to this immune synapse. So you need the channel, and you need the channel.
Yeah, no, of course, ori will be recruited there
for proper generation of the structure. What I think is that we just happen to have an experimental system with these activating cover slip that sort of maximize the defect. That's my feeling.
I mean, we've seen ori localizing to the IS as well. I'm just surprised that you have such a strong effect on the T cell receptor. So that I found surprising.
Yeah,
but it's clearly there.
I mean, some of the reviewers suspect that this may be kind of an artifactual nature. So what they want is they want us to make a more physiological synapse. So for that, we'll just use Reggie cells and have a cell-to-cell interface. And I think maybe the data are different there, but given the defect that we have here, I'm quite sure we'll find something there. I'm sorry, I cannot find the questions. Is there a list of the questions?
Yeah, there's the chat function where people are writing the questions, but you can also, everyone, if you just want to ask a question, just turn on your microphone. I don't see anything more in the chat function. So if you want to ask something, just shoot ahead, and especially, all the younger ones, and just turn on your microphone and ask. It's good to grill Nick a little bit.